Good afternoon, everyone. Dr. Minal Bhujangra Wagmar is here. In the series of diseases of field and horticulture crops and their management, we are going to see our next crop, that is mustard crop. Coming to the first disease of mustard, very important disease, that is Alternaria Blight of mustard. Causal organism Alternaria brassicae. In this, we can see that dark brown spots appear on the lower leaves, and this spots is covered with the brownish margin around it, and that concentric rings get appear around the spots, and the spots are called as a target spots. Inside these spots, this area get dry and necrotic appearance occur inside the spots, and leaf shows the necrotic appearance. Some chlorosis also appear inside the leaf. Lack of chlorophyll will be there. Ultimately, it reduces the photosynthesis rate and down the yield. In this, our oil content of the crop and their quality get damaged or reduced, and also shrivel the seeds in the infected pods. And if this disease is appearing at early stage, it showing some damping of root get rotting kind of root shows rotting kind of symptoms in the early stages, and damping of kind of appearance also occur inside this disease. Coming to the life cycle, majorly two important spores are formed. That is, chlamydosaurs is a resting or overwintering spores in this life cycle, and secondary infection is continued by the conidia spores, and conidia spore will continue the secondary infection with the help of rain and wind and other factors like tools and etc. Coming to the next disease, that is white rust of mustard, causal organism Albigo candida. It is a oomycetes disease. It comes under the oomycetes fungi. In this, we can see pustules get appear on the lower side of the leaf. That is, creamy whitish pustules will develop on the lower leaf of the surface. And once these pustules get collapsed, like merged together, it forms the patches on the lower leaf. In this, also, that leaf get damaged due to this pustules formation and lead to the less photosynthesis rate, lead to the yield losses. In this also, oil content of the crop get damaged and size of seeds also shriveled. And coming to their life cycle of disease life cycle, we can see that it shows the uh, two kind of spores. In this, antheridium, that is a male gametangia, and oogonium, female gametangia, fuse together and they lead to the formation of the oosphere. First they form the oosphere, and then it due to the gametangial contact, they finally lead to the formation of oospores. Once this oospores undergoes the germination, it lead to the form of the sporangium structure, and this sporangium structure after rupturing, it lead to the zoospores. Zoospores are kidney shape in structure and biflagellated. Like this, two life cycles are happen inside this disease. Sexual life cycle continue by the oospores, and sexual life cycle continue to the zoospores. Sexual life cycle continue under the favorable condition, and under the unfavorable condition, a sexual life cycle is continue. These zoospores get disseminated from one place to another place. With the help of this flagella, they can swim in the rain splash. Coming to the next important disease, that is Downy Mildew. Causal organism is the Pernospora brassicae. Again, this is a oomycetes fungi. In this, yellow and pale yellow spots appear on the upper leaves. It also shows some chlorosis, lack of chlorosis, and whitish purplish downy fungal growth, and appear on the leaf underside. It shows the motile and chlorotic appearance to the leaf. In this also, important spores are, that is oospores and zoospores. Here also, Thredia, male gametangia, and Oogonia, female gametangia, due to gametangial contact lead to the formation of first oosphere, and that oosphere form the oospores. Oospore having a very thick wall. Here it has a very thick wall structure. So it can act as an restrict spores inside the soil debris, and it can survive for years to years. And again, when host is available with favorable condition, it will germinate and continue to the life cycle and lead to the infection. Otherwise, this undergoes the germination, and indirect germination it formed to the sporangium, and the sporangium structure having zoospores. Once sporangia is break, the zoospore release have its kidney shape and biflagellated. This can swim and disseminate from one place to another place with the help of rain splashes. With the help of this flagella, they can locomote from one place to another place. Coming to our next disease, and it's the last disease, important disease of the mustard. Cause that disease is a Sclerotinia stem rot. Causal organism Sclerotinia sclerotiorum. In this also, typically water-soaked lesions or spots appear on the leaves, and later it converted to the whitish cottony growth on the leaf surface. It also shows some chlorosis appearance on the leaf, and lead to the lack of chlorophyll, and lead to the reduced rate of the photosynthesis. In this, size of seeds get shriveled, and also reduce the size of pods, and oil content also reduces. In this, basically, hard structure, that is sclerotia, is formed inside the infected stem pith. This is a blackish color hard structure. We can find if you see the mustard crop is infected by Sclerotium stem rot disease, we can find this hard structure sclerotia inside the soil or in a stem pith. And coming to their sexual life cycle is related by the ascospores. Ascospores is released by the apothecium fruiting structure. It is a cup-shaped fruiting structure. It releases to the ascospores. It leads to the sexual life cycle of these diseases. And typical conidia is not produced in this disease. Coming to the combined management of these all diseases, definitely we should follow some good cultural practices such as deep summer plowing and crop rotation, because due to crop rotation, continuous mustard crop availability for this all Alternaria Blight, Downy Mildew and stem Sclerotinia stem rot, and for white rust, continuous host availability is not available. Due to this, pathogen will not able to continue their life cycle in next season also. And due to crop rotation, host is not available, the life cycle of the pathogen is breakdown, and like this way we can control the pathogen on the plant. Again, some coming to the chemical fungicide, carbendazime and captain and thyrum we can use this for seed treatment, and trichoderma viride also, and bacillus subtilia as a biological control. In this, mainly in mustard, downy mildew and white rust, we use for the mancozeb and metalaxyl and copper oxychloride spraying, and mancozeb and metalaxyl combine also goes for the seed treatment of the 3 gram per kg and plus 3 gram per kg. And if we are separately spraying only mancozeb and metalaxyl seed treatment, we go for the 6 gram per kg rate for seed treatment. We also can do the foliar application of the metalaxyl, mancozeb and copper oxychloride at the 0.25%, that is 2.5 gram per liter. First spray we can do in a when initial symptoms are appear, and second spray after the 15 days we can do for another spray. If there is no need of second spray, we will not provide the second spray. So these are some important management, mainly metalaxyl and mancozeb plus copper oxychloride. We provide mainly to these mustard diseases for downy mildew and white rust, and for other diseases this thyrum and carboxyl and our carbendazime are provided as a fungicide management, as a seed treatment as well as foliar spray. And we can also do some other cultural practice, that is sanitation. Distort the infected plant debris to reduce the inoculum. So like this we have covered all important diseases of the mustard. If you like this video, please like it and subscribe my channel and share my video. Thank you.